Hi everyone, and thanks for joining us for our extended educational webinar behind the scenes of the development of a COVID-19 vaccine suitable for global access. I'm Lucy Lawrence, the scientific content producer here at Technology Networks, and I'm here to moderate the event. I'm delighted to be here today to bring you some great talks from experts in the field. And at the end of each talk, there'll be a Q&A session, which gives you the chance to engage with our speakers. So I'd very much encourage you all to ask questions, as this is the best opportunity to get answers from our speakers. I'll explain how you can ask questions at the start of each presentation. Please do make sure you navigate over to our literature section, where you can download the latest resources from the sponsors of today's webinar, Thermo Fisher Scientific and ACROBiosystems. I'm pleased to introduce our next speaker, Nicolas Abuid, with his talk. A very warm welcome to you, Nicolas. Before we begin, I really wanted to share some of Nicolas's background with you. He is the West Coast Field Application Scientist for ACROBiosystems. He's a trained biomedical engineer whose doctor focused on the type 1 diabetes cell therapy. But at ACROBiosystems, Nicolas is working with leaders in both industry and academia to support their research needs. He's part of a team that helps to guide in the use and development of recombinant proteins that can be used for COVID-19, T-cell therapies, as well as many other immune and autoimmune conditions. As a member of ACRO, he's highly interested in leveraging the company's resources and state-of-the-art technology to help their partners and clients develop next-generation therapies. It's set to be a fantastic presentation, and following the webinar, we'll be hosting a Q&A session where we welcome any questions that you may have for Nicolas. Remember that questions in this webinar can be asked at any time, during or after the presentation, and even if you're watching this on demand. If you're wondering how to ask questions, you can enter them into the text box on the left-hand side of your screen and press send. Any questions that we don't have time to answer today, we will ensure we contact you offline with an answer. If you do come across any technical issues during the presentation, please click the question mark in the top right-hand corner to request our support. I'll now hand over to Nicolas.
Thank you very much for that kind introduction. And as mentioned, my name is Nicolas Abuid, and I'm an application scientist for ACROBiosystems. And today I am here to present on the critical role of protein reagents in vaccine development. And with that, I would just like to give out a little introduction about this pandemic and some of the many efforts that we went through to kind of control the spread of the virus. First, we started with increasing the manufacturing of PPE. Then we moved towards imposing either a social distancing mandate or a mask wearing mandate. And as the pandemic progressed, we were obligated to look into certain different therapeutics, such as remdesivir, to treat hospitalized patients for the treatment of COVID-19. This was out in August of 2019. Then we looked into dexamethasone as a well-known and effective anti-inflammatory for hospitalized patients with COVID-19. And finally, monoclonal antibody treatment for this virus. However, as history has taught us, the main and only solution for pandemics is a vaccine that can help stop the spread of the virus, as we have seen with the polio vaccine, the MMR, the smallpox, and the flu shots. With the COVID vaccine, we saw also an increase in vaccination rate that came with a decrease in the number of cases. However, the COVID-19 can also prevent infection and decreases the chances of serious illness and mortality, but also prevent the long-term side effects that persist even after a mild version of the vaccine. And it can prevent the spread and mutation rate of this virus. Because of the importance of vaccination, several researchers and leaders in academia and industry have focused their efforts in developing a vaccine for this virus. There are 7 well-known strategies for vaccine development. Some of the most well-known is the inactivated or lab-attenuated vaccine, as shown as commercialized by the BBIBP, Sinopharm, and Coronavac, Sinovac. Also, we have the protein subunit vaccine that was developed in the US and is commercially known as Novavax, and the RBD vaccine, or the baculovirus SF9. There's other approaches, such as the virus-like particles. This approach has had better luck with HPV and other types of diseases, but for the COVID-19, we know that the virus vector commercialized by Janssen Pharmaceutical in the US, or the commonly known AstraZeneca in the UK. And one of the most widely used in the US is the mRNA and Pfizer vaccines or the Moderna and Pfizer vaccines for mRNA strategy. And similarly, we also have DNA vaccines known in the US by Inovio and Insightus in India. Regardless of the strategy you use to develop a vaccine or the different approaches you might take, every researcher has to follow a certain line of work to get to the end goal, which is develop a vaccine. First, we start with production and quality control. Then we move to clinical trials, preclinical studies, and then we move to clinical trials phase 1 and 2, which leads to clinical trials phase 3. In ACRO, we've had many ways in which we contributed to this workflow, and starting by the development of, or the expression of a spike protein and ACE2 protein, which are two interactions known to help spread the virus in the host. This interactions this protein can be used to study these interactions, and on top of that, we help and contribute with the studies by providing protocols for ELISA, SPR, BLI, and flow cytometry. In addition to providing these studies and the protocols, we also have a very rigorous quality control to maintain the purity and lot-to-lot consistency of our products. And we also provide SDS-PAGE, SEC miles, HPLC, just for our collaborators to have well-documented quality control reports for the proteins that they use. In addition to the various proteins expressed, we've gone an extra step and developed some sandwich ELISA kits to quantify the content of the antigen. We developed kits to detect SARS-CoV-2 spike protein in sample, as well as RBD titer assay kits and spike protein ELISA kits for vaccine development. In addition, we in ACRO are always working hard to stay up to date with the different mutations and variants of concern that are coming out, and we're trying to have a repertoire of all these proteins with different mutations to assist researchers in their studies and vaccine development. Following, we are also working in efficacy evaluation, which we do this by developing ELISA kits used to detect the titer of anti-SARS-CoV-2 mouse IgG antibodies in serum plasma in vitro, and detection of mouse anti-SARS-CoV-2 antibodies in serum in vitro as well. We have developed competitive ELISA kits for detection of the titer of anti-SARS-CoV-2 neutralizing antibodies, and we provide the positive controls, which is an anti-SARS-CoV-2 spike RBD neutralizing antibody. This antibody can be paired with other anti-SARS-CoV-2 spike S1 antibodies to detect SARS-CoV-2 spike S1 protein in either a sandwich ELISA or an LFA assay. Once the QC and production stage is concluded, we can move on to preclinical studies. For this, we have kits indirect ELISA kits to detect the titer of anti-SARS-CoV-2 monkey IgG antibodies and antibodies, anti-SARS-CoV-2 spike RBD neutralizing antibodies that can be paired with other spike S1 antibodies to detect a spike S1 protein in either an ELISA, a sandwich ELISA, or an LFA assay. And as I mentioned before, we have rigorous QC, and we provide reports such as the SDS page among different lots, among different batches, to ensure that our collaborators have a very well-documented set of proteins and reagents. So once the preclinical studies is over, we can move to the clinical trials phase 1 through 3. In this area, we work on ELISA kits used to detect the titer of anti-SARS-CoV-2 human IgG antibodies, which we developed for the titer measurement of anti-SARS-CoV-2 antibodies IgG, spike RBD in human serum, a spike trimer in human serum, as well as serological tests for IgG titer of anti-SARS-CoV-2 antibodies in serum plasma in vitro. And on top of this, we also have a wide range of the human IgG subtypes antibodies for serological tests of IgG titer of anti-SARS-CoV-2 antibody in serum plasma in vitro, and antibodies, specific neutralizing antibodies against SARS-CoV-2 spike protein RBD domain with minimal to no cross-reactivity against SARS-CoV spike protein. And with this, I would like to move on to some examples that in which we have collaborated with different vaccine developers. First, with the SARS-CoV-D, where these researchers used recombinant S1 spike protein to develop the DNA vaccine candidate known as the SARS-CoV-D. In these studies, recombinant S1 spike protein was used to study the antibody response after DNA vaccination in biopsy mice, guinea pigs, and rabbits, where all these rodents were immunized with a vaccine construct, and serum was collected after a certain period of time and evaluated for SARS-CoV-2 as one specific IgG antibodies. And finally, I would like to talk about the how we collaborated with the Pfizer vaccine studies by providing a recombinant SARS-CoV-2 RBD, which was used to study the robust immunogenicity that was acquired from the vaccination with the BNT162b1. This RBD binding IgG concentrations were detected 21 days after the first dose, and this was substantially increased 7 days after the second dose given at day 21. So with this, I would like to move on to any questions and that you might have for me or my colleagues.
Thank you very much, Nicholas. I'm sure you'll all agree there were some excellent points raised during the presentation. So yes, we will now begin the Q&A session. And to submit a question to any of our speakers today, you should enter your question into the box on the right-hand side of your screen and click send. We'll answer as many questions as possible during the time we have available. We've had several questions submitted already, so I'll move straight on to these. The first question we've had in are, what are some efforts done by ACRO to keep up with the new mutations and variants?
Thank you very much. So our team is continuously learning about the new variants of concern and their incidence. And as we learn about this, we develop products to assist the field in studying these variants, mutations, and help develop vaccines for these different coming mutations and variants that are a threat to the end of the pandemic.
Brilliant. And the second question we have here is, how often do you QC different batches or lots? And how are some of the ways that you collaborate with different vaccine developers?
Oh, thank you. So our quality control team is diligently testing every batch and lot to make sure our collaborators are ensured consistent results in the product. So we are aware of the obstacles and challenges researchers in academia and industry face, and we try to do our best to minimize some of the obstacles by keeping high quality products with consistent purity and sterility, especially in the toxin. In addition to this, we do provide products. We try to constantly meet with customers who might seek customized products and learn from their requests. And we have a team of scientists that are eager to learn and develop these new products and variations of the existing products, and speed the development of new technologies, treatments, and help this pandemic come to an end.
Mm-hmm. Absolutely. And another question we have is, has ACRO ventured, or does it plan to venture, into the diagnostics field to control the pandemic?
Yes, actually, several of our customers and potential collaborators are working on novel diagnostic techniques, but our role in this line of products is just providing either proteins or kits, and keeping the high quality products to expedite their testing, but not specifically with diagnostic technologies.
Mm-hmm. And how can the technology that's been developed during the pandemic be used in development of vaccines for other diseases or conditions?
Actually, many of these technologies in vaccine development were commercialized and translated to clinical stages. So these technologies will have great implications for treatment of diabetes, other viral diseases, and there's like great work that can help create immunization for diseases that didn't have a vaccine.
So, with the pandemic coming to an end, what has your team learned, and how can we apply what you've learned to rapidly address or control the future risk of pandemics?
Thank you very much for this question. So scientists in the field are rapidly innovating and working on different treatments and therapeutics to not only find cures for existing diseases, but control disease spread of new coming diseases. And we have a wide range of technologies that are ready to be applied, and our team of scientists can always help with developing products for QC, for development of not only therapeutics but also immunization. And in future pandemics, if any, we'll be rapidly able to control this and control this and be able to handle it in a timely manner. So as our technology increases, we're able to address these diseases and new conditions in a faster and faster way. And yeah.
Fantastic. Well, thank you very much, Nicholas. Unfortunately, they're all of the questions that we have time for today. And just to remind everybody, any questions that we didn't have time to get answered today will be answered offline as soon as possible. And you can continue to ask questions even when watching this webinar on demand.